Hi, this is Miss Litton, and we are reviewing APBio. Say hey.
Hey.
All right, so we have already done Big Idea 1. Big Idea 1 is about what? Evolution. Big Idea 2 is about energy, growth, development. Big Idea 3 is about what? Pick a key word out of there.
Respond.
Information, yeah. How we store, receive, transmit, respond to information. So the big ticket item there is information. So let's go through. First one, 3A, heritable information provides for continuity of life. What heritable information are they talking about? Genes, DNA, good job. We already know central dogma. I'm not even going over that again because that's, you got that, stone cold pilot. We know that there was some debate about what could be the genetic information because we know how many nucleotides do we have? 4. But how many amino acids do we have? 20. So it seemed like that we could make more combination with 20 letters versus 4 letters. Yes? You with me on that? Okay. But there were several different experiments that were done. We knew it had to be, it had to store information, it had to be stable, and one more thing. Starts with an M.
Mutation.
Mutate. It had to be able to mutate, right? Why do we need it to mutate?
To get variation.
To get variation. Good job. All right. So we looked at different historical experiments where they were looking to see that DNA is in fact our code book. So you remember about Griffith and the mice and 2 different strains. One was lethal, one was not. And what was the experiment that was done? Oldest bio or blue seat tell Grace sleep something. Tell them about what the experiment was.
So you had to strain S
killed S, makes it kill the mice.
and that was like something.
That was
Okay, does this help you?
Yeah.
He killed S, and then they took the heat killed S. He killed S, so it should not have done anything, but they mixed it with live R. What happened to R? You did it. What is it? Transformed. Just like you transformed your bacteria, right? You hopefully transformed your bacteria to give them antibiotic resistance, and you also gave them other DNA that you had genetically engineered. That is a transformation. Is that a vertical transfer or a horizontal transfer?
Horizontal.
Horizontal. Vertical means parent to offspring. Horizontal is you're picking it up from your environment. All right? Okay. Next, Avery, McLeod, and McCarty, they proved by using enzymes that digested proteins versus digesting DNA that it was in fact DNA that did the transformation. We know all about this, Hershey and Chase and bacteriophage, and we knew that they injected something in there. Either, remember, Hershey and Chase didn't have the structure of a bacteriophage, but they knew something was transforming those organisms. Now we know all kinds of things about viruses, right? What are some additional things you know about viruses now? Maybe like their life cycle? I'm just curious what those are. Lytic and lysogenic. Which one do you become a prophage in? Lysogenic, right? That's when you hide in the bacterial chromosome. Yeah? Integration, good. And so they did their experiment with an isotope. This would be easy pickings data for them to give to you on measuring the amount of an isotope before and after, and from that you would infer that the transforming agent was DNA, right? So remember they put an isotope on the protein. Remember what that was? S35. And then on the nucleic acid, P32, base sugar, phosphate, right? And they went to see what isotope was found in the bacterium. And they found that it was P32, so they knew that was the transforming agent, was a nucleic acid. I think that's all on here, yes. Okay, structure of DNA, Watson and Crick, a double helix. They used evidence that was maybe borrowed, also gifted. And one of the things was shining X-ray through DNA, and the X-ray through the DNA showed that it had a what kind of structure? A helical structure, and there was a repeating pattern. What was another piece of data they used to come up with the structure of DNA? Yes?
The
Chargaff, right? Matching however many A's you have also, T's, and G's you have C's. They also knew the components of DNA, and then they knew bond angles. And so they knew they could come up, that's how they came up with the double helix structure. Okay, there's Chargaff's data, Rosalind Franklin. Okay, now, we learned a long time ago that proteins can make enzymes, and enzymes, and all proteins for that matter, are very, very what? Specific. Remember that part? And so we know things like when you do DNA replication, right, that the enzyme can work in one direction, but the DNA, right, is anti-parallel. It runs two different ways. When I say the DNA runs in two different ways, what am I referring to? We identify those the leading and lagging strand, but it's the way the sugar is arranged, right? And so we can identify the carbons in every single sugar molecule. Carbon 1 is always the one connected to the nitrogenous base. And then you work your way around. If you think of it like a house, you can't go on the roof in that order because up on the roof isn't oxygen. Got to go down, right? So we can see here, here's carbon 1 on the sugar, 2, 3, 4, and a chimney makes 5. There's an oxygen right there, even though you can't see it. So if you're an enzyme looking at this, what you're looking at is a 3', but if you were looking in this direction, you'd be looking at a 5'. And right across the street, this is 3', and this is 5'. And DNA polymerase 3 can read from 3 to 5, making complementary 5 to 3, right? And so if you go at one point, okay, like you stand in the middle of a 2-lane street, cars are going to be coming towards you from 2 ways, and cars are going to go away from you in 2 different ways. Do you agree?
Yes.
Okay, so 2 of those strands would be leading, and 2 of those strands you'd have to backstitch in, and that would be the lagging strand. Okay, regardless, to start the party, you need to have RNA primase lay down a little bit of what? Primer for DNA polymerase to work off of. On the lagging strand, primer needs to be put down multiple times for that replication fork as you're working your way out. Okay, you have to keep putting down that primer so there's something, you have to backstitch because you can only travel in one direction, and so you have to keep backstitching. Okay, now, do you remember that we said the primer has to be removed? What's the name of the thing? Who is the one to save us? DNA polymerase 1 removes the primer and replaces it with DNA nucleotides. So the RNA nucleotides come out, DNA nucleotides get replaced, and then what do you use? Ligase, which you're familiar with because you did ligation with it in your lab. All right. So if you look at a structure of DNA like that, you know what all the components are, yes?
Yes.
Okay. Here's DNA replication. It's referred to as being semi-conservative because each old strand serves as a template for new. Perfect. Okay. Oh, and there it is, DNA replication. Take a look at it, see if anything's scary, if you have any questions about it. Okay, moving on. So right here, we're moving out in both directions, right? So you could have a leading and a lagging strand as you move out. Remember, eukaryotic chromosomes are going to be linear, and prokaryotic cells are going to have what? Circular. We know the differences between DNA and RNA. The difference is what? What are all the differences? The sugar. One single-stranded and one is double-stranded. What else? Uracil is found in RNA, and thymine is found in DNA. And don't say exactly the same, or remember it's complementary is the word you want to use. It's complementary. Adenine is complementary to thymine, right? Okay, or adenine is complementary to uracil, right? So that's who it can form a hydrogen bond with. All right, on the genetic code, the central dogma, DNA to DNA is called what? DNA to DNA is called replication. Using the DNA to form RNA is called transcription. Now there's 3 RNAs you could form, only one of them will be translated, right? You have mRNA that will be translated, but you also transcribe rRNA and tRNA. Yeah, they're just not translated. You're all right with that? Okay, and the mRNA is going to get and every 3 bases on the RNA is called a
Codon.
which is complementary to the anticodon located on the tRNA. Okay, and remember the different spots on the ribosome? Okay, yes. Okay, so you have your A site, you have your P site, and your exit. Okay, so those are your different kinds of RNAs. We went over that. When we talk about post-transcriptional controls, we can talk about mRNA processing. What are the things we do to process our mRNA before it leaves, which you don't have to do in a prokaryotic cell, can immediately be translated. Literally, while transcription is taking place and the mRNA is hanging off, the DNA, right? Because there is no nuclear envelope, no barrier. It can immediately get translated. They don't have what? What's something they don't have besides a nuclear envelope? What else what doesn't prokaryotic DNA have? No, it has transcriptional control. It's usually negative. Yes? Yeah, it only has exons. Yeah, no introns. Okay, only exons that are expressed. They don't have the capability to snurp out the introns. Yeah, they don't have that. So that's why if you move a eukaryotic gene into a prokaryotic cell, you have to give it a clean copy with no introns in it, only exons. Okay, that right there could be an essay on why you need to do that. Okay, how could you do that, by the way? Use reverse transcriptase on your mRNA that's already been processed?
Yes.
Okay. And then get DNA, and then you'd use something like replicase to make it double-stranded, and then you insert it in there. Right? Duh, everybody does it. All right. So introns and exons, we talked about that. We know that, right? That's easy peasy. There's some tRNA, anticodon. These are different loops, right? There's some parts where it's complementary to create those loops. Okay, Mata and Nuremberg gave you your genetic code. Remember, what did we use to predict the amino acid sequence? We used the mRNA. Now, if you know the DNA, you can know what the mRNA is, or if you know what the tRNA is, you know what the mRNA. You always look up the mRNA. That's the genetic code. And there's more than one way to get several of the amino acids, right? There's 3 stop codons. There's 1 start codon. The start codon is always what? AUG. And it's always your first amino acid is methionine. Now, why wouldn't every single protein have methionine on it, part of it, if it is the initiation codon or start codon?
Because not every.
If you don't need it, why would you need it?
Well, no, if every single protein cannot be formed unless you do the initiation codon, which would give you methionine as an amino acid, which would then make it as a component of your protein. How come every protein doesn't have methionine in it if that's what you need to start translation?
Not everything is needed by the cells in that particular.
Post-translational control. Post-translational control. You will remove some parts, you can cut away some of the amino acid of the protein you built, and then you remember the chaperone proteins folded together? So as a part of post-translational control, you may remove some segments of those proteins that are not in the final form of that protein. Just like when you make mRNA, you have your initial transcript. That may not be what you use to build your protein because you're going to cut out the introns. You can cut out pieces of the proteins in order to make your final protein product. Okay? That's why you can have one gene and multiple proteins from it due to both post-transcriptional control and also due to post-translational control. Got that? That's a great essay question. They should ask you that. Okay? Because you have to understand it. All right. We know all this. We know this. Did you know that?
Yeah.
I wonder why I felt the need to put that, but I felt it. Yeah, let's just talk about that. Genotype codes for phenotype. All right, recombinant DNA. That's our total business. You've been doing recombinant DNA for 2 weeks, so I think you probably feel pretty good about that. You use plasmids as a vectors, but you could have used, what's another good vector? viruses. Okay, you know about sticky ends, right?
Yes.
You know how to do cloning because that's what you did. PCR, what does PCR stand for? Polymerase chain reaction. Why do you need to do why would you need to do that? Multiple copies of DNA. Maybe you want to do some testing, some analysis, some identification, right? To do that, it requires a thermocycler, which is a series of heating and cooling reactions and you sprinkle in some nucleotides, you put in some primers, you put in some TAC, right? Remember TAC? Thermus aquaticus, harvested from archaeans, right? And they could take the temperature fluctuations. You use their DNA polymerase 3, so you get multiple copies of your DNA. Gene amplification. There it is. Okay. DNA fingerprint. You did some DNA fingerprinting, right? Because you could look at your gels and go, I have a specific fingerprint I would expect for PCR, or sorry, I don't know where I was coming from with it. Pecan, Piara. There's a certain fingerprint you would expect if they were cut appropriately. That's a DNA fingerprint, right? Where you're comparing them. Next. Now, this is there's an error in their model right here, but they're using tandem repeats. So in our DNA, we have things where it stutters in the DNA. It might go ATA, 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 ATA. And in the chromosome I got from my mom, let's say chromosome 7, I might have 50 repeats of ATA. And in that area from my dad, maybe I have 20 repeats, right? So there's a difference there. So when I cut that DNA up, one has a really long section of that ATA and one has a shorter section of that ATA. Do you agree with that? Do you understand that? Check, check. Okay, so when I do gel electrophoresis, I could get two different banding patterns. If I have the same number of repeats, I would get the same banding pattern. What's wrong with this picture, though? Analyze it because you've done gel electrophoresis. What is it? Tell me, everybody. Yeah, they flipped this, right? This should be the 16, so this one should be out here, not back here, right? You can analyze that because that doesn't line up correctly. That's super disappointing. Gel electrophoresis, you know all about that. Those are bioreactors to grow up genetically engineered bacteria. You gave bacteria a red fluorescent protein from a eukaryotic cell. Do you now understand when I say you could have made it make clotting factors, right? You could have made it make a hormone. You could have made it make an antibody, and all we'd have to do is lyse the cells and harvest that protein from it. Just like if you've checked your cells over there, if you got red fluorescent protein, that protein belonged to what kind of organism? Sea anemone. So why, if I can take something from a sea anemone and get a bacteria to get it, why couldn't I take something from a human and get it, right? Because we're both eukaryotic. Does that make sense? Okay. A genetic profile, this is when they do probes to identify, and they'll take single-stranded DNA, and then they will have single-stranded DNA like on a computer chip, and then they will denature your DNA and see what sticks. And if it matches, it means it's the same gene, and you have that, could be a problem, you know, if they have some mutated DNA that they're matching you to. Gene therapy, ex vivo, in vivo. In vivo means what? Inside of you. Ex vivo, you remove your cells, you fix them, and then you return them. Yes? Oh, crazy baby. Okay. So in sexual reproduction, that form, when you do that, the information that you pass on in sexual reproduction is from two different sources, right? Whereas if it's asexual reproduction, it's from one source. Good job. All right. Look, it's the cell cycle. G1, what happens during G1?
Grows.
Yeah, cell grows, organelles increase. What happens during the S stage?
DNA synthesis.
DNA synthesis. Oh, there it is. DNA synthesis. What happens during G2? Protein synthesis. Prophase, metaphase, anaphase, telophase, cytokinesis, right? And telophase and cytokinesis may overlap. Is cytokinesis uniform in all cells? No. Where would it be different? Plant has a cell plate, which becomes a cell wall. Or a fungal cell could have one. Or a fungal cell could actually be multinucleated. So there's variations in that process. Controlling the cell cycle, very big deal. Could be external signals, could be internal signals, which turn that cell cycle on and off. We talked about checkpoints. I said cyclins are like tickets to get on a plane, and then the checkpoints. are areas where they make sure that the cell is progressing appropriately through the cell cycle. If not, what are you going to do to that cell cycle?
Apoptosis.
Yes. Well, first you'll try to fix it, and then if you can't, then you will kill it. Fix it first, then kill it. Programmed cell death, we talked about that yesterday, I believe.
Yeah.
Or was it the day before yesterday? It's all
Either
stringing
one.
together for me right now. Histone, non-histone proteins. These are important, okay? Histone proteins, remember the nucleosomes where you spool the DNA to organize the chromosome? Okay? Those are probably not regulatory because they're all the same. Non-histone proteins, this could be related also to epigenetic inheritance, right? You could have the trait, but it's not expressed because you don't have access to it to even do transcription. So those could be your non-histone proteins. Meristematic tissue is continuing. In plants, this is going to be asexual, right? For us, we have somatic cells in our bone marrow that will continue to develop. Stages of mitosis, I'm pretty sure you feel confident about that. Homologous pairs, same size, same shape, and code for the same characteristics. Who is going to have homologous pairs, sexual or asexual organisms? Sexual. Prokaryotic or eukaryotic? Eukaryotic, because our chromosomes are linear, right? Now, who doesn't have homologous pairs in this room? Boys. Yeah, males. Right? Why?
Yeah, Y and X.
Yeah, why? Because of the Y. Right? Why? Right, because your 23rd pair is not XX like all the ladies in the room. Okay? It's XY or YX, right?
I would always say sorry you're a boy because that's
The Surrey gene. Yes, I like it. I like it a lot. Okay, you know that one. I mean, there. Okay. Now, this makes me think about gene linkage. What's a way to break up gene linkage? Crossing over. Gene linkage, which of Mendel's laws does gene linkage?
Oh no.
Independent assortment, right? Independent assortment says I can get big Q with big R, big Q with little r. But if Q and R are on the same chromosome, I can't have that, right? They're linked together. Where big Q goes, so does big R, if that's the linkage, right? The only way I can get big Q with little r is if what occurs? The likelihood of crossing over, it is more likely if they are farther apart or closer together. Farther apart, because you have more opportunities to do that crossing over. If they're close together, it's very hard to do crossing over, right? Because it limits where you can do that crossing over. Okay, meiosis. The focus of meiosis 1 is to separate the homologous pairs. The focus of meiosis 2 is to separate the sister chromatids. Good job. I'm going to reiterate again. Watch your Bozeman videos on the science practices. Watch it on the new AP exam. He does a big, huge review. It's like 40 minutes or something like that. Those are really good reviews to be like, do it early in the morning, like a Saturday morning, and go, okay, I'm going to do this now, and be ready to go on it. Those kind of reviews can be very, very helpful. Again, I'm going to remind you, go to the College Board website. I think you'll do fine on the multiple choice. Do you not feel like you'll do fine on the multiple choice? Right? If you bother to read it, bother to answer it. Speed, right? Read, focus, and remember, you'll be able to write all over those exams. That will help you stay focused because you're going to write on those exams. I just don't have the paper for you to do that. Okay, write on those exams. The part that you want to see is you want to look at the essays and you want to look at try to write them, but study the rubrics. Look to see how they grade the essays. That should not be a foreign thing to you, right? That was such a good use of time. Just go, remember from 2013 on. I don't even know if they have the older ones up there anymore, but it's 2013 and on. Those are the essays you're going to want to look at, right? Those, because they've released all the essays for all the exams from 2013 and on. Okay? All right. Homologous pairs separate in meiosis 1. Sister chromatids separate in what? Meiosis 2. Good job. Okay, we know that, we know that, we know that. That's easy peasy. Comparing mitosis and meiosis, not a problem. Why are these puppies different? 3 reasons. Independent assortment, fertilization, random sperm and eggs, and crossing over. Good job. Okay, so we know that. There's crossing over, independent assortment, random fertilization. Okay. Prior to Mendel, it was the idea of blending. Is there anything that looks like blending, though?
Yes.
What's that called?
Incomplete
dominance. Now, there's another one that's kind of like incomplete dominance. What is that? Codominance. Okay? In codominance, you get to see both things. You get to see the red and the white. In incomplete dominance, it's just they're layered on each other, so it appears to be pink. Yeah. Then let's not forget about epistatic genes. Epistatic genes, you may not be red, you may not be white or pink if the epistatic gene says you're not going to lay down any color, right? Because that switch has to be on. You might have white albino rats, but if you're big B, they're black rats. If they're big B, little b, they're brown rats. But you're not going to get black or brown unless you have the coloring gene, and that is getting expressed. Okay. Yeah. You can do crosses, yes?
Yes.
That's not a problem. That's so easy. We've done it. Oh, okay, good. Okay. Now, if there is more than one way to get the same thing, right? And you just think about a Punnett, if you think about a Punnett Square, right? You want to multiply, if it is the exact same thing, big T, little t, big T, little t, little t, big T, right? Those ultimately will get you the same thing. Do you agree with that?
Yes.
Okay, so on those, you need to add up that because that will be the same thing. There's more than one way to get the exact same thing. If you could have different outcomes, right, in sequence of events. So when we talked about, let's say, flipping a coin, what are the odds you're going to get a head? Half. And what are the odds that a second coin is going to be a heads? Half. And what are the odds that a third coin is going to be a heads? So that all three simultaneously come out heads, you'd have a half times a half times a half. 2 times 2 is 4 times 2 is 8. So there's an eighth of a chance they all turn out heads. Right? Okay? But if I talk about one of them could get heads, tails, one could be tails, heads, right? So that's when I say, well, that's when I can start to add them up, right? Because they're going to come out the same way. That's like your Punnett Square. Big T, little t, big T. All right. Test cross. Why would I ever need to do a test cross?
To see if it's even possible to get, like,
when I have a dominant trait with an unknown genotype. So I have the dominant phenotype, okay, but I have an unknown genotype. Emily has brown hair. I don't know if she's brown or brown blonde. Who could I mate her with?
blonde.
There. Ethan? All right. So, because I know he will every time give a blonde, because he's recessive, right? If they have 10 children, okay, 10 children, then we can probably assume that she's homozygous brown. But if they have one blonde child, what do we know? Heterozygous brown. Okay? So here is purple. And by the way, you may not do that because I know you're thinking about it. Just kidding. Oh, Chisquare test. Okay.
Do they ever give Chisquare as the...
Yeah, you totally. That's one of your equations, right? Did I not give you math practice problems? Yes, I did. Remember, you are the last year of Britons.
Yeah.
Right?
Oh
now, keep in mind.
So hard for next. I hope it's really hard.
Next year to get rid
No.
No.
Fewer essays and fewer questions.
They're getting like 53 multiple choice questions.
We better get like a curve.
Yeah. Now,
we
okay,
can
you can't do anything about that now. Just listen.
I hate letters.
Okay, listen to me. A grid-in is worth just as much as any multiple choice question. I think this is why they're getting rid of them, because they're like, skip the grid-ins and make sure, at least you have a chance of getting one right, because one of the answers is right on a multiple choice, right? You have to pull that out and calculate it for a grid-in. If it's not going to take, if you're trying to decide, go with your time, right? Some people go, do all your grid-ins first. Well, I don't want you to spend an inordinate amount of time on a grid-in when it's worth the same amount as any other multiple choice question. So don't spend any more time on a grid-in than you would any other multiple choice question, right? Because at least a multiple choice question, you have a chance at getting it right, right? Because it's going to be, one of those answers, right? Okay, but Chi-square test, it might be on your essay, right? They might give you data, and then you have to analyze that data, right? So chi-square test is one of the easiest of all the tests, right? Because all you're doing is taking what did I see over what did I expect? Missed expectations, okay? How much am I off, right? Square it and put it over what you expected. Do that for every single data point, add it up, and see if it falls within what's acceptable, right? And so we accept 0.05, right? That is okay. But anything after that, if we look at our degrees of freedom, remember, on the chart where you look, if you have two choices, then if it's not one, it's the other. That means you have how many degrees of freedom? 1. If you have 3 choices, like red, white, pink, and a cross of flowers, that's 3 choices, so you have 2 degrees of freedom. If you have 4 choices, round and yellow, round and green, wrinkled and yellow, wrinkled and green, how many choices do you have there? 4, so you have 3 degrees of freedom. Okay? And what are the odds that you're going to get that? This number will be closer, right? This number, if you're getting really close to what you would expect, then that number should be very, very small. If you're really off from what you expected, that number is going to be really, really what? Big. Okay? It's going to be really, really big. And then you just have to see, was I predicting I would be close to the number, or was I predicting I was going to be far from the number, and look to see if that helps support your statement, right? What was your prediction? Did you expect your data? Most genetic crosses, you expect it like 1 to 2 to 1, right? Or 3 to 1. And that's what you think it should turn out. So if you get a big number when you're predicting 3 to 1 or 1 to 2 to 1, that's not a good thing, right? It means something else is coming into play. But if you're trying to see how does sleep affect your preparation for the AP exam, right? And you're thinking, I think sleep has, you remember when we talked about null hypothesis? Null hypothesis is status quo. So you're looking for a difference, for it to be significant, right? You're looking to see, hey, there was a big difference in AP scores between those that were sleeping and not sleeping, so sleep must be significant because the data was so far off the null. Yeah?
So if you have your null hypothesis, then when you get your chi-square and the number's really big,
then that would support, remember, because the null hypothesis, it's kind of like a backwards way, right, of supporting your point. Because the null hypothesis, you're saying there is no effect on sleep in the AP exam.
Okay, so if the number was really big, would it be significant?
Yeah, then you would go, well, sleep has to be a factor. You can't say it directly, but you can say at least sleep was a factor, if that number's really big.
Is it fine to put it in a if then because format, or is it just a statement, this is not connected to this?
You can look to see what they're looking for.
Okay.
Hypothesis you generally want to do if, then, because, but look to see what they're after on that question.
For say, if the exam said put in a hypothesis and I put if then because, and it has the same meaning, but theirs was just directly, this is not connected to this.
Well, that's if you're doing a null hypothesis.
Yeah.
So you have to look to see. They'll probably take it either way, and I don't know that you could probably format that in a way that you could say, if I was giving a null hypothesis, I would say this, If I'm doing a research hypothesis, I would say this, right? A null hypothesis is HO, research hypothesis H1. Like H0, H0, that's like no connection, Yeah.
So if you're
That's
doing a
hard.
chi-square test and you get like 0.01, then there's no change, right?
Right, then your data was exactly as you would have predicted.
Right. Okay.
Yeah. Okay. We did this experiment in class. You can do those practice problems if you would like. Hello, my friend.
Hi, I have a question, but I'll ask you later.
Okay, is it a quick one?
It is quick, but it's not important. So
Oh, okay.
I'll talk to you later.
Okay. Okay, we've done this. Okay, so look right here. Here's your degrees of freedom. Okay, so these numbers right here are big, right? So if I'm trying to say, hey, I expected this data and I didn't get it. Oh, what's going on here? Okay, if you get a big number, look at these big numbers. Those are saying then, that's a very poor fit, right? Smaller numbers mean I'm very close to what I had expected. Unless you're doing, let's say, a null hypothesis and you want a big difference because you're trying to make a point, right? You're trying to make that connection. So look to see how it's being used. It's just two sides of the same coin, right? right? A null hypothesis is just about the opposite of a research hypothesis. So if it's a research hypothesis, you want your number to be very, very small. If it's a null hypothesis, and you really think it does have an impact, then you want it to be big. You okay? Check, check, check. All right. Incomplete dominance, we know about that. This is another example. Straight hair, curly hair, hybrid makes wavy. Blood types. What are blood types a really good example of?
Codominant.
Codominance. Yeah, and where would we see that? AB blood. What's another good thing that blood is good for? Multiple alleles. You have big I A, big I B, and little I. So it's not like you just have tall, short, round, wrinkled, green, yellow. Okay, in this case you have big I A, big I B, little I, which means you can get more combinations of genotypes, which means you can have more resulting phenotypes, right, from that, okay? And there's more than one way to get type A blood, right? So if we look at type A blood, you can be big IA, that's type A. This would be like a hybrid type A blood, right? And then type B, same thing. AB, there's only one way to do that. And then type O. Let's talk about our immune system. What's the best blood to have if you need blood all the time?
AB.
AB. AB what? AB positive or AB negative?
AB negative.
AB positive. That means you will not attack A blood, you will not attack B blood, you're definitely not going to attack O blood because there's nothing to attack, and you won't attack the RH factor, the positive, right? If you want to be I want to be the best blood donor ever. What do I want to be if I want to be the best blood donor? O negative. Because I don't have A, I don't have B, I don't have RH. Yes?
When it means positive or negative, what does that mean?
That's the RH factor. RH factor. That's just another antigen on the blood. It's a third
Type
antigen.
O blood also accepts all of it, right?
No.
Oh, no.
No, it doesn't. Okay, look, you guys, let me just finish this. We're 5 slides away from the end of this Enduring Understanding. Okay, pleiotropy. We talked about that, right? When one set of DNA has multiple effects, you could talk about Down syndrome, and the GART gene has multiple effects affecting the way your brain processes, the structure of your hands, structure of your mouth. Polygenic inheritance. Polygenic, what does that mean? Many genes. So that means like your intelligence is not set with just one homologous pair of genes, right? You have A's, B's, Q's, R's, whatever, giving you your intelligence. Height, skin coloring, all of those kinds of things. And then environment. The environment can have an influence because something in the environment could create like epigenetic inheritance, right, on you, where it could influence it that way. Temperature is one. Temperature for the way the bunny rabbit's hair grows in, and sex determination of reptiles. Oh, there's the sex determination of reptiles. Nature versus nurture. How many of our traits, and we had a whole thing on that, right? Traits that were determined by your environment versus traits that you were in your DNA. Remember like snake and the way snakes tasted slugs?
Oh, the flick of the tongue.
Yeah, the nesting, how they built their nest with long strands or short strands, that was innate. But where is behavior due to the environment? Imprinting,
Like the
right?
ducks.
Associative learning, the ducks, right? All of those are where the environment influences it. That's it. Good job, you guys. Got one done. You did how many slides? Yeah, you completed 97 slides. Well done. Well done. Well done. We'll start there tomorrow. Have a piece of toast. Absolutely. Let's finish it tomorrow.
That's gonna be like
You're super smart.
140 slides.